So before breaking down the Bernardi and his associates and collaborators' evidence and conclusions to this article, we first want to get into what is this mystery theory behind the non-coding DNA. What this non-coding DNA mystery refers to is the idea that much of the human genome is comprised of junk DNA, or some even refer to it as selfish DNA. And what they mean by the selfish DNA is that it kind of acts as like a molecular parasite attaching to a host and has no real adaptive behavior. And the junk DNA referring to the idea that is a non-coding protein sequence within the genome. This idea that there are regions of the DNA that only pass down to offspring for the sake of DNA. Essentially, there's this idea that roughly 98% of the DNA of human DNA is actually this junk and doesn't have serve a real purpose. But after recently they've found studies, thanks to Bernardi and his collaborators, that this junk DNA actually serves a real purpose. For instance, it works in gene regulation. With tRNA, rRNA, and regulatory RNA, as well as it plays a role in forming a nucleoskeleton, serves as an attachment site for meiotic apparatus, and functions as a mutation buffer, and also dictates the overall 3D architecture of the chromosomes. So now getting into the evidence of what Bernardi and his collaborators and his team have found. He looked at the genomic code, which is the fundamental link between DNA structure and chromosome structure. And his team have actually developed evidence on that isochores reflect the fundamental unit of chromosome organization in chromosomes domains. For instance, H isochores are guanine and cytosine rich, or GC rich, euchromatin, which have an oligo G sequence, which basically weakens the binding site of topically associating domains. And on the contrary, L isochores are GC poor, heterochromatin, and they refer to this idea of gene deserts. These two isochores actually refer to that are involved with the change to the structure or mold of the 3D structure of chromosomes. Now, breaking down this article, the main thing that stuck out to me was that there was this astounding idea that 98% of the human DNA is considered junk DNA, all because it doesn't code protein, which I found just unreasonable. How no one has questioned what does this actually do, or maybe we just didn't have the technology developed yet that led us to this point of what possibly could this this junk DNA that seems repetitive actually perform. And I think with within recent years, due to technology advancements, along with the study of the Human Genome Project, it's clear cut that this is a huge barrier wall that they've been able to break through. And I think moving forward, figuring out what this junk DNA and the non-coding mystery can resolve is going to be tremendous for our medical advancements as well as our biological advancements moving forward. That is all, and thank you.